Hello everyone, my name is Alec, and my research project investigated modifying Bacillus subtilis to assist lead bioremediation of Ulupoloi. So I was inspired to do this project because I've been fascinated with utilizing genetic editing, a kind of biotechnology, in real life. However, I ended up learning about the ethics of using biotechnology instead. What is biotechnology? Well, biotechnology is the field of using living systems and organisms to create or produce products that benefit humanity. And technology in this field includes genetic editing, which is the modification of DNA sequences to express desired traits in an organism. One such tool for genetic editing is CRISPR, and that stands for clustered regularly interspaced short palindromic repeats. And this essentially involves using bacterial immunity systems as a pair of scissors to cut out specific sequences of DNA from any organism's genetic code. Other DNA can then be inserted into the organism to replace or add sequences to the genome. Well, what are the concerns of genetic editing tools? Well, for instance, genetic editing can be used for therapeutic purposes, like treating genetic diseases. However, it can also be used for non-therapeutic enhancements, such as creating designer babies that are smaller, taller, or stronger than normal. And so a real-life example of something borderlining this is the 2018 incident where Chinese scientist He Jiakui edited human embryos to have HIV resistance and was subsequently arrested and sentenced to prison for this disregard of ethics. Additionally, in recent years, there's been a rise in a movement towards accessibility of biotechnology. Previously, using biotechnology required expensive machines that only university-level laboratories could afford. Now, people can buy kits to perform CRISPR in their own garages. This leads me to my first research question for this project. It asks, what are the ethical concerns that arise from allowing genetic editing to be performed by average people, such as high schoolers like myself? It's important to note that genetic editing is not all negative. It has lots of utility to solve problems. For instance, genetic editing has been proposed to edit mosquitoes carrying avian malaria that can threaten Hawaiian honeycreepers so that mosquitoes cannot carry the disease. Another proposition is to use CRISPR to revive the woolly mammoth, an extinct species from the Ice Age. Therefore, the problem I wanted to consider is lead contamination in Ulupoloi. This lo'i is located in Kawainui Marsh that was once a taro patch, but then became used as a landfill site for several decades. Recently, the site has been cleaned up by several nonprofit groups with the goal of restoring it for growing food crops. But testing has revealed that there are traces of lead contamination in the lo'i, and that prevents its safe usage, since plants can absorb lead, and that makes them toxic. A previous independent research student researched how different bacteria tolerated and mobilized lead from the lo'i. And in addition to this, hyperaccumulators, which are plants such as Indian mustard that can uptake high amounts of metals, were planted in an attempt to absorb significant amounts of lead from the lo'i. However, the lead was not taken up by plants from the lo'i in high enough quantities for it to be safely used. And one hypothesis for this could be that the lead was stuck to the soil of the lo'i and therefore could not be uptaken by the plants. I researched and discovered that bacteria can be used to remove lead. Bacteria have previously been used for biomining, which is the extraction of metals and minerals from ores. And bacteria also have several ways to extract and tolerate toxic metals like lead. For instance, they have extra and intracellular precipitation. This reduces toxicity and allows higher concentrations of lead phosphate to build up. Additionally, through binding by specific proteins known as metallothioneins, which are proteins involved in protecting the cell against toxic metals, and biotransformation of lead compounds, which transform lead compounds into volatile forms to diminish their toxicity. Additionally, they can bind lead through siderophores, which reduces the concentration of lead. Therefore, I propose that a possible solution could be to genetically modify bacteria to assist in modifying the lead and mobilizing lead from the taro patch. So, can bacteria be genetically modified to remove lead from the lo'i? Well, bacteria can be genetically modified, to, yeah, with the most notable example being the insertion of the human insulin gene into yeast cells to produce insulin. Using modern technology, a vast library of information about biological systems can be utilized in designing and modeling a system digitally, before even attempting to perform the experiment in real life. I've decided that the specific requirements for a bacterial chassis for this project would be that the bacteria must be found in Ulupalogi, as well as having a fully sequenced genome, so I can decide where to make edits. This brings me to my third research question. What organisms should we be allowed to edit, and for what purpose should we be allowed to edit organisms? An initial candidate for genetic editing was a bacterial strain known as Dechia ulupuensis. This bacteria filled out both of the mentioned requirements. The bacteria is native to the Loi and is associated with the sacred pink edible mud of the Kwai Nui Marsh. And this mud is actually a bacteria-like yogurt mix, and is associated with the legend of King Kamehameha I when he was invading Oahu. While he was at the marsh, he and his troops ate the mud when there was a taro shortage, and it gave them enough strength to win the war. For this reason, it holds cultural significance to Native Hawaiians. So, when I asked the question, should K61 be edited, I decided that K61, since it holds cultural significance to Native Hawaiians, it would therefore be unethical to modify something that a culture values. Instead, I found an alternative candidate in Bacillus subtilis. Bacillus subtilis also fills out the specific requirements of being found in the Loi and having its full genome sequenced. Additionally, it has been thoroughly studied and has the potential to be a part of many available biological parts. It is easily integrated with foreign DNA as well, and this makes it a suitable candidate for this project. In conclusion, after analyzing the ethics of editing different bacteria, I've concluded that ethical concerns are dependent on who is using biotechnology and for what purpose it is being used for. Bacteria can be edited to help mobilize lead from the sour patch. However, before editing any organism, including bacteria that people may not care about, one must just consider the impacts of such actions, as seen as in Dechia ulupuensis. In this project, I learned that I should not be afraid to ask for help. Researching is researching new information, and is a new experience for myself as well as other researchers. I also learned that research is a trial and error process. You will not get everything right on the first try. It took me many different attempts to achieve the project that I have today. Some advice that I would give for future independent research students are to find passion in your project. This way you will enjoy what you're doing and will not think of it as a chore. You will also need to learn time management. Keeping up with the project through different papers and presentations is tricky and cannot be crammed up in the night before it's due. Finally, commitment is necessary. By taking this class, you're making a commitment to a research project, and you must complete it. I'd first like to acknowledge my friends and classmates, as well as my family, for supporting me throughout this entire year and project. I'd also like to thank Mr. Eric Tong for helping me in accessing many of the research documents for the research. And finally, I'd like to thank my mentor, Dr. Chan, for supporting me through the whole project.